Okay, guys, it's lab 4, aseptic technique and streak plates. This lab is going to teach you how to move bacteria from one place to another without contaminating the sample, and also the way that we do get our isolated colonies on agar by streak plating. So um, we call it inoculation. So those colonies we grow on the agar, if we need to move them, for example, if we have several different types of bacteria on the same agar and we use our colony morphology to tell them apart, we may want to move one colony to a completely new agar plate so that it would be by itself and uncontaminated. So we would take a little metal loop, we would put it into the fire really quickly, make sure that it turns red, and then give it just a few seconds to cool down. Then we would take it and we would use the loop to get part of the bacteria off the one agar plate and then take it over to the next agar plate and inoculate it. There's a little bit more to it than that. I'm just trying to give you a thumbnail on this one. The problem with this, and this is something that in the actual lab some folks have trouble with, is getting this done without actually getting contaminants. If you'll recall back from the ubiquitous microbes lab, the air and everything around us is full of bacteria. So in order to do this, we have to be very careful, especially not to wave our loop around and pick up contamination, because we need a pure culture in order to do those biochemical tests. If it's mixed, then the results will be just so much garbage. So aseptic or sterile technique is how we make these pure cultures so that we can characterize them. We use sterile media and equipment. So all the tools in the actual lab are put in an autoclave and sterilized prior to use. And we do use our Bunsen burners to sterilize the tip of the loops to make sure that they are free of any other bacteria. Flame sterilization. And when we move bacteria from one liquid culture in a test tube to another, we also, as we pull the top off that test tube, we run the flame, the opening of that test tube near the flame so it will kill any bacteria that are in the immediate area. Most bacteria you're going to find in nature is a mixture of all different types. And so that makes it hard to be able to run the chemical tests to determine what it is. So we have to separate this, and we use the streak plate method. And streak plate is a solid dilution technique. In streak plate, let me give you a brief overview. So we flame the loop, cools down, we get bacteria off one plate, take it to the next other plate, and we do this zigzag thing. Okay, we lift it up, we carry our loop back to the flame and flame it again, and we let it cool, and then we go back, not to the original agar plate, we're finished with it. We're going to go to the one we already have, and remember we did these zigzags. We're going to do zigzags at a 90-degree angle. What are we doing? We're not adding any more bacteria, and we flamed it after we put the first batch on here because we want to spread out the bacteria, not add more. If we add more, then they're all going to be crowded. And instead of having individual colonies, you'll just have a bacterial lawn. It'll just look solid. So anyway, we do that second streak at that 90-degree angle. When we finish there, we go and we flame our loop again. And again, we got to let it cool down. I say this, guys, because these guys are live. And believe it or not, if you put a red hot loop on them, they will fry. How about that? And this happens in the lab frequently. So we let it cool for a second or two. I usually do a 10 count before I do it. And then go back and we did the first loop in this direction, second loop in this direction, and then we're going to come down here and we're going to actually get on that second zigzag, and then we're going to pull like this. And I usually open up the zigzags a little bit so that there'll be a little bit more space. And then of course after that we go ahead and flame the loop because we're finished with it. So in the actual lab they were going to give you a liquid culture of E. coli and a plate culture. You would take your Bunsen burner, and the striker is the way we light those guys. We were going to have 2 culture tubes, those of BHI, brain heart infusion. That's just sterile liquid that has nutrients in it so the bacteria will grow. And the TSA plates, tryptase soy agar, we talked about those before. When you adjust the Bunsen burner, you do it so that you actually see 2 flames. So you'll see the normal yellow flame, but in the center you'll see that blue flame. And this light blue area at the very top. And that top portion of that blue flame is going to be the hottest part. So you can see it on here on this diagram that they've got that. So there's the wire loop. They run it through, and it's good to angle it down like that so that you get as much of that wire in there. And I usually keep it in there until I see the metal turn red. And then once that's happened, then I move it away from the fire without waving it, because if you wave it, you're going to contaminate it. And just give it a chance to cool so I don't kill the bacteria I'm trying to transfer. Now here's what we were talking about, flaming the mouth. So they've already taken the top off, so they've got the very top near that, not in the flame, but near it, so that that air just adjacent to it will be sterilized. So apparently they've already flamed this loop. They've gone through this procedure. They go down, and just like we talked about with a solid where we actually get a colony, here we just let the loop get a loop full of liquid. We take this out, we flame it again, and then we put the lid back on. When we do that, I don't know if you guys may or may not be able to see, use these 3 fingers, the little finger and the ring finger and the middle finger, to hold the top of the test tube. You hold that in the same hand that holds the loop. It sounds really hard, but it's really easy to learn and get used to. So you flame it, and as soon as you're finished flaming it, you get that tube on there. Because the longer the tube is off, the more chances we have of contamination. The same thing with the tops to the agar plates. I only keep it off just long enough, and I actually just tilt it up a little bit. I don't take it completely off, and I get that thing closed as soon as possible. If you leave it exposed to the air, then the airborne bacteria get in there, and they're going to undo all your hard work. Okay. I think these are just the step-by-steps of what I told you before. So that would be, once you get it out of the broth like that, then you can do 1 or 2 things. You can do the same thing. You can put it on a TSA plate. See, there's our first loop, like so. Then you go and you reflame it, cools down. Then you come over here and you grab some of that one zigzag and you go for the second at 90 degrees. Then you go and flame it again and come for the last one. Yeah, I'm sorry, they've turned it upside down. It's actually this way. They just turned the picture upside down, but it's the same thing. Now, as far as doing, that's a broth to plate transfer, where you take bacteria from a liquid culture and put them on a plate. To remove them from a broth to a broth, liquid to liquid, is very simple. Once you have that bacteria in the loop, you simply grab the other tube, flame the opening as you pull right after you pull the top off, and then you stick the loop in there and swirl it around, and that's a broth to broth. Plate to plate transfer or plate to broth. To do a plate to plate is simply exactly the same. You just get a loop full of that solid colony, not too much. Less is more in this situation. And you do the same 3 streaks with flaming in between each of the streaks and getting no more bacteria from the original plate. And in a broth to plate or plate to broth, you simply take it off and put it in the liquid and swirl it around. It's very simple. And they've got all the instructions down here, guys. These are pretty good. I've seen this, and this one especially I want you to go to. That is going to allow you to actually simulate doing a streak plate. And that's what we call these. These are streak plate method. That's how we get pure cultures. 2 reasons why the aseptic technique is important. Define pure culture, purpose of streak plate. You guys already know all the answers to this. All right, guys, that's lab 4.